गुड इवनिंग एवरीवन, वेलकम टू बायो कैटलिस्ट। तो आज हम लोग सेल साइकिल एंड सेल डिवीजन स्टार्ट करने वाले हैं। तो चलिए शुरू करते हैं। सेल साइकिल। Cell division is a very important process in all living organisms. During the division of a cell, DNA replication and cell growth also takes place. All this process, that is cell division, DNA replication and cell growth, hence have to take place in a coordinated way in order to ensure correct division and formation of progeny cell containing intact genomes. The sequence of the events by which a cell duplicates its genome synthesis that occur constitutes of the cell cycle and eventually daughters into two daughter cell is termed as a cell cycle. ये definition हो सकता है, ठीक है। Although cell growth in terms of cytoplasmic increase is a continuous process, DNA synthesis occurs only during a specific stage in a cell cycle. The replicated chromosome DNA are then distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. Phases of cell cycle. A typical eukaryotic cell is illustrated by human cell in culture. These cells divide in approximately 24 hours. However, this duration of the cell cycle can vary from organism to organism and also from cell type to type. Yeast, for example, can progress through the cell cycle which is about 90 minutes. Okay? Oh no. Which is about 90 minutes. The cell cycle is divided into two phases, interphase and M phase, mitosis phase. Okay. M phase. The M phase represents the phase when the actual cell division or mitosis occurs. When the actual cell division or mitosis occurs, and the interphase represents the phase between the two successive M phase. It is significant to know that in the 24-hour average duration of the cell cycle of the human cell, cell division proper lasts for only about an hour. The interphase lasts more than 95% of the duration of the cell cycle. The M phase starts with nuclear division corresponding to the separation of the daughter chromosomes, that is karyokinesis, and usually ends with division of cytoplasm, which is cytokinesis. The interphase, through which the resting phase, is the time during which the cell is preparing for division by undergoing both cell growth and DNA replication in an orderly manner. Interphase is further divided into three phases, that is G1, S and G2. G1 phase corresponds to the interval between the mitosis and initiation of DNA replication. During G1 phase, the cell is metabolically active and continuously grows but does not replicate in its DNA. S or synthesis phase makes the period during which the DNA synthesis or replication takes place. During this time, the amount of the DNA per cell doubles. Okay, if the initial amount of the DNA is denoted as 2C, then it increases to 4C. However, there is no increase in the chromosome number. If the cell is diploid or 2n number of chromosome at G1, then even after S1 phase, the number of chromosome will remain the same, that is 2n. In animal cell, during the S phase, DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm. During G2 phase, proteins are synthesized in the preparation for mitosis while cell growth continues. Some cells in the adult do not appear to exhibit division, that is hurt cell, and many other cells divide only occasionally as needed to replace the cells that have been lost because of the injury or cell death. These cells that do not divide further exit the G1 phase in order to enter an inactive stage or quiescent stage, that is G0 of the cell cycle. Cell is the stage which remains metabolically active but no longer proliferate unless called on, so depending upon the requirement of the organism. In animals, mitotic cell division is only seen in the diploid somatic cell. However, there are few expectations to which where haploid cell divide by mitosis. For example, male honeybees against these plants can show mitotic division in both haploid and diploid cell. Now let's come to M phase. This is the most dramatic period of the cell cycle, याद रखना ये part, involving a major recognition of virtually all components of the cell. Since the number of the chromosome in the parent and the progeny cell is the same, it is also called as equational division. Through the convenience of the mitosis has been divided into four stages: nuclear division, karyokinesis. It is very essential to understand that the cell division is a progressive process and very clear-cut lines cannot be drawn between the various stages. Now, karyokinesis involves the four stages, that is prophase, metaphase, anaphase and telophase. Let's study about this. Prophase, it is the first stage of the karyokinesis of mitosis, follows the S and G2 phase of interphase. In the S and G2 phase, the new DNA molecules formed and are not distinct but intertwined. Prophase is marked by the initiation of condensation of chromosomal material. The chromosomal material becomes untangled during the process of chromatin condensation. The centrosome, which had undergone duplication during the S phase of interphase, now begins to move towards opposite poles of the cell. The complication of the prophase can thus be marked by the following characteristics of the events. Chromosomal material condenses in order to form impact meiotic chromosome. Chromosomes are seen to be composed of the chromatids attached together of the centromere. Centromere, which had undergone the duplication during the interphase, begins to move to the opposite poles of the cell. Each centromere radiates out microtubules called as the esters. The two esters together with the spindle fiber forms the mitotic apparatus. Cell at the end of the prophase, when viewed under the microscope, do not show Golgi complex, endoplasmic reticulum, nucleolus and nuclear envelope. Metaphase. The complete disintegration of the nuclear envelope marks the start of the second phase of the mitosis, hence the chromosome is spread through the cytoplasm of the cell. By this stage, condensation of the chromosome is completed and they can be observed clearly under the microscope. This is then the stage at which morphology of the chromosome is most easily studied. At this stage, metaphase, chromosome is made up of the two sister chromatids which are held together by the centromere. Small disc-shaped structures at the surface of the centromere are called kinetochores. These structures serve as the site of the attachment of the spindle fiber of the chromosome that are moved into the position at the center of the cell. Hence, metaphase is characterized by all the chromosome coming to lie at the equator with one chromatid of each chromosome connected by its kinetochore to spindle fiber from one pole and its sister chromatid connected by its kinetochore to spindle fiber from the opposite pole. The plane of alignment of the chromosome at metaphase is referred to as the metaphase plane. The key features of metaphase are the spindle fiber attached to the kinetochore of the chromosome and the chromosomes are moved to that of the spindle fiber and equator aligned along the metaphase plate through that of the spindle fiber of the both plates. Anaphase. At the onset of the anaphase, each chromosome arranged at the metaphase plate is split simultaneously and then the two daughter chromatids, now referred to as the daughter chromosome of the future daughter nuclei, begin their migration towards the two opposite poles. Each chromosome moves away from the equatorial plane. The centromere of each chromosome remains directed towards the pole and hands at the leading edge with the arms of the chromosome trailing behind. The anaphase stage is characterized by the following key events. Centromeres and chromatids separate and centromeres move to that of the opposite poles. Important, okay? Now let's move to telophase. At the beginning of the final stage of karyokinesis, that is telophase, the chromosomes that have reached their respective poles decondense and lost their individuality. The individual chromosome can no longer be seen at the each set of the chromatin material tends to collect at each of the two poles. This is the stage which shows the following key events. Chromosome cluster at the opposite spindle fiber poles and their identity is lost as that of the discrete elements. Nuclear envelope develops around the chromosome cluster at each pole, forming the two daughter nuclei, and nucleolus, Golgi complex, and reform. Now, cytokinesis. Mitosis accomplishes not only segregation of duplicated chromosome into daughter nuclei, but the cell itself is divided into two daughter cells by the separation of cytoplasm called cytokinesis, at the end of which that cell division gets completed. In an animal cell, this is achieved by the appearance of a furrow in the plasma membrane. The furrow gradually deepens and ultimately joins in the center, dividing the cell cytoplasm into the two. Plant cells, however, are enclosed by a relatively intaxable cell wall. Therefore, they undergo cytokinesis by a different mechanism in the plant cell. Wall formation starts in the center of the cell and grows outward to meet the existing lateral walls. The formation of the new cell wall begins with the formation of a simple precursor called the cell plate that represents the middle lamella between the walls of the two adjacent cells. At the time of the cytoplasmic division, organelles like mitochondria and plastid get disturbed between the two daughter cells. In some organisms, karyokinesis is not followed by cytokinesis, as a result of which multinucleated condition arise, leading to the formation of the syncytium, that is liquid endosperm in coconut. Is that clear? Now let's move to significance of mitosis. Mitosis or equational division is usually restricted to the diploid cell only. However, in some of the lower plants and in some of the social insects, haploid cell can also divide by mitosis. It is very essential to understand that the significance of the division in the life of an organism. Are you aware of some examples where you have studied about the haploid and diploid insect? Answer in comments. Mitosis usually results in the production of the diploid daughter cells with the identical genetical component. The growth of the multicellular organism is due to mitosis. Cell growth results in disturbing the ratio between the nucleus and cytoplasm. It is therefore become essential for the cell to divide in order to restore the nucleocytoplasmic ratio. A very significant contribution of mitosis in cell repair. The cells of the upper layer of the epidermis, cells of the lining of the gut, and the blood cells are being constantly replaced. Mitotic cell division in the meristematic tissue, the apical and the lateral cambium, result in the continuous growth of the plant throughout their life. तो आज हमने पूरा माइटोसिस के बारे में पढ़ा एंड उसके जो जो डिविशंस है उसके बारे में भी डिटेल में एक्सप्लेनेशन के साथ पढ़ा है एनसीईआरटी रीडिंग का। तो जितना तक अभी पढ़ा है वो घर में रीडिंग करना और जहाँ जहाँ मैंने हाईलाइट किया है वो भी देखना, क्योंकि नेक्स्ट क्लास में इसके ऊपर ही एमसीक्यू लाया जाएगा। तो अच्छे से ये वाला पार्ट देख लेना एंड नेक्स्ट क्लास में हम लोग मियोसिस का पार्ट कंप्लीट करने रहेंगे। ये चैप्टर ज़्यादा बड़ा नहीं है तो शायद 2-3 क्लासेस में ही कंप्लीट हो जाएगा। तो जितना आज पढ़ाया है वो अच्छे से रिवाइज़ करना, क्योंकि एमसीक्यू लाएंगे कल। तो आज के लिए इतना ही। फॉर नेक्स्ट मोर क्लासेस फॉलो द चैनल एंड सब्सक्राइब। थैंक यू फॉर नाउ।